know of 1 hour of silence from the construction, which fortunately you didn't hear, but those of us who've been in the building a while did. So we better take advantage of that hour and go ahead and start. We're very pleased to have as our speaker today, Bonnie Bassler from Princeton University, who's going to talk about quorum sensing, bacterial quorum sensing.
Yeah, thanks, John. Yeah, I'm happy to be back this morning to give the technical seminar, which is really about what my lab's working on, one of the projects my lab's working on right now. And I just want to say I've had an absolutely magical trip. So thanks for letting me come give all of these lectures. It's really, really been fun. Okay, so I'm going to sort of pick up where I left off yesterday. And if you didn't come yesterday to the lay people seminar, don't worry, because I have 3 overlapping slides, which is really all you needed to hear yesterday. So let's see. So I want to talk today about how bacteria tell self from other, and then how that lets them have high fidelity communication. And so to get there, so let me start with just a little bit of overlap from yesterday. So at the heart of what my lab studies is how bacteria get any bang for their buck, right? So what we know historically about bacteria is that they do all of these terrible things like cause illness in humans and plants and animals. Increasingly, what we're learning is that bacteria are essential for life because they grant us all of these functions that human genomes don't encode, right? So they live in and on us in a consortium all throughout our lives. This is called the microbiome. And what these beneficial bacteria do are things like digest plant food, educate your immune system to keep these invaders out, provide micronutrients. We're only starting to learn about all of these fabulous jobs that the bacteria that are always living with us take on, basically on our behalf. And so now we get that bacteria have 2 kinds of personalities, right? They're these terrible guys that can kill us, and then the superheroes, these miniature superheroes that keep us alive. But then the question that my lab has been asking for the last 20 years is, irrespective of whether you want to think about harmful behaviors or beneficial behaviors, how do bacteria get any bang for their buck? They're so small, how can it be that they can do these kinds of tasks? And so what we've shown is that the way bacteria manage to do a lot of these big jobs is by communicating with one another with a chemical language, by organizing their gene expression so that they behave like multicellular organisms to carry out tasks in synchrony. And when they do things together, they can accomplish these feats that they couldn't accomplish as individuals because they're too small individually to make an impact on the environment. And so this process of group behavior in bacteria is called quorum sensing. And at the simplest level, the way we think about bacteria now is that they live in 2 different modes. They're either living alone and carrying out individual behaviors, or they're living in communities and they're carrying out group behaviors. And the way they modulate between those 2 modes is by communicating with these small molecules that we call autoinducers. So the way that it works, and this has been shown in hundreds and hundreds of species of bacteria by now. And so the way that it works is when bacteria are at low cell density. So the world is really big, the bacteria are really small, right? So they want to have the program of gene expression going that's good for carrying out solo behaviors. So they're doing some subset of tasks that they are capable of doing, but among the things they do is that they make and release small molecules that you can think of like hormones, but we call them autoinducers. And so what happens is when the bacteria are at low cell density, the autoinducers diffuse away, the bacteria can't detect them, that sets the program of gene expression to tell the bacteria to carry out individual behaviors. But then as they grow and divide, right, since everybody is producing its share of autoinducer, the concentration of the chemical increases in proportion to cell number. And when that chemical hits a particular threshold concentration, the bacteria interpret, when they encounter the chemical, that they must have neighbors around, that they're now at high cell density. And so when they detect the compound in unison, all of the bacteria change their gene expression programs and they begin to carry out collective behaviors, so group behaviors that takes a lot of bacteria working together to make the task successful. And so the 2 sort of biggies that have been that I talked about yesterday and that the field is, gaga about, is that they make biofilm, so how they sit on surfaces and carry out these sort of surface-bound community activities, and then also the group-wide expression of virulence factors. So we now get in pathogens that they have to have quorum sensing or they can't be pathogenic because they don't understand to launch these cascades, or they only launch these cascades when they're in groups, right? Because that's the effective way to carry out a successful infection. So the field is sort of moving toward that, and that's what I talked about yesterday, but I want to actually go back to the origins of this field. And it was all discovered, the reason I drew the bacteria in blue is because the original group behavior that was discovered, that where quorum sensing came from, is that bacteria, this was from marine bacteria, and they turned on bioluminescence as a group. So what we could show is that when they were alone, they didn't make light. When they were all together, they turned on bioluminescence. And what's spectacular about bioluminescence is that you can measure it quantitatively over 4 orders of magnitude. So it was this amazing readout that we could see and we could measure a bacteria acting as a group. And then we knew we were right, because, and we're going to do this trick today, is because what we could do is spin the cells out and collect the cell-free supernatants. And so the molecule is in the soups, right? And so if you add the molecule to dilute cells, they will turn on light. Right? And so we knew that there were these molecules involved. Okay, so this all came, and so I'm going to use bioluminescence for the entire talk as the readout of quorum sensing. And so then the bacteria there were 2 bacteria that this started with, and one of them is called Vibrio harveyi, which is this free-living bioluminescent bacterium, right? And this is just a picture of what we measure in the lab, right? You turn the lights off in the room, the bacteria at high cell number, quorum sensing has kicked in, and they make light. Right? And so we can measure that, either with your eyes or actually with machines. You can measure it carefully, right? But what I want to say is that light production is what I'm going to measure as the symbol of the bacteria talking to each other. But Vibrio harveyi, like every other quorum-sensing bacterium that's been discovered or been studied, it's much more than one process. So light is the thing we can see, but what we know in Vibrio harveyi from doing microarrays and other kinds of technologies is 600 genes turn on when the bacteria kick into quorum sensing mode. Bioluminescence is just one of the traits. Most of the traits that turn on in all quorum sensing bacteria are what you call public goods. So I already told you about making a biofilm, expressing virulence factors, exoproducts, so enzymes that chew up solid substrates. So most often when bacteria turn on quorum sensing, they're giving something away to the world, right? An exoproduct. And then the entire group shares it collectively in that product. But there's a problem with that, is that when you give it away, it can be exploited, right? So the question is, how do you do quorum sensing? How do you manage public good production without getting cheated on? And that's what I want to talk about. You really want to give these products away, but you only want to give them away if your kin get them. Okay, I'm getting hot. Right, so in the ocean, that was yesterday's talk. In the ocean, almost everything, like 75% of everything, either makes or uses somebody else's light. And so in the ocean, you don't have to go very deep, and there's no light. And so nearly all the organisms, so light's a big deal in the ocean. The light is blue, which is what travels really far in the water. And so many Vibrios that are closely related to Vibrio harveyi are symbionts. And so they give an animal light, and the light uses it to find a mate, to attract predators, to escape from prey. And in exchange, the bacteria gets fed, and they make many more progeny. And so that's sort of mostly what bioluminescence is used. This guy's free living, and so it's more of a puzzle, like why it makes light in the free living state, right? But the light is selected to be seen, right? Because it's what travels far. Okay, so I think that's a really curious, question that my field has wondered about since the discovery of this. What's key for today is that they only turn on light when those molecules are there, right? When they're talking to each other. Okay. Okay. All right. So this is the readout that I'm going to use. Okay, so what we've shown is that these molecules that I'm showing you on these cartoons, that Vibrio harveyi, this free-living bacterium, actually uses 3 different autoinducers that we discovered to communicate. So the first molecule is this one. It's called a homoserine lactone. We call this autoinducer 1 because it's the first one we found. And so what you can see is that it has a ring and then has a side chain. And today you have to actually learn some of the chemistry. It has 4 carbons on the side chain, and there's a hydroxyl group on carbon 3. So this molecule, as far as we know, is only made by Vibrio harvii. We've never found another bacterium that makes that molecule. So Vibrio harvii makes and detects it. And so we interpret that to mean that this molecule allows intra-species communication. This is how Vibrio harvii counts its kin, its clones, its identical twins. The second molecule we showed Vibrio harvii makes is this one. It's a fatty acid, and we call it CAI1, right? And it's 13 carbons long. And what we showed is that all Vibrios, every Vibrio that's been sequenced and that's been studied, makes a molecule that looks that is either this one or looks a lot like this one, and the receptors are a little bit sloppy, so they detect each other's. And so we think that this molecule is for intragenera communication. This is how Vibrios know each other. But the gene that makes the enzyme that makes this molecule exists in no other bacteria except Vibrios. But every Vibrio has it. So we think that's about the family of Vibrios, right? So this molecule says you're my twin. This molecule says you're my cousin. So then the third molecule that we showed that Vibrio harvii makes is this 5-carbon molecule that I talked about yesterday. We call it Autoinducer 2 because that's the order we discovered them. And this molecule is made generically in the bacterial world. So gram-negatives, gram-positives, bacteria that live in all kinds of different niches, and they all make the identical molecule. So we think this is the bacterial trade language. This is for interspecies communication, right? It just says bacteria. And so what we think then is that the way, and there's probably more molecules to discover, but what we think these molecules do is that they encode 2 things. First, the concentration of them encodes something about cell number, right? So the more there is, right, because they're building up as the cells grow. So it says something about how many cells are in the vicinal community. But then the other information that's encoded in these molecules, beyond how many, is something about who is that? Is it me? Is it somebody that's related to me? Or perhaps it's the enemy, right? And so those are the way we're starting to think about quorum sensing as we flesh out the lexicon that these bacteria talk with. Okay, so those are the molecules. And then we also have figured out what's the sensory system that allows the bacteria to detect the molecules and integrate the information to turn on and off genes. Okay, and so I have a very simplified diagram of the circuit here because I'm only showing you the proteins that you need to know about today. So there's 3 autoinducers, right? And they're outside the cell, and they each have a dedicated receptor. So we call them, everything's basically Lux, for Luxor, the god of light. So LuxN, LuxPQ, and CQSS. So these are the 3 receptors. And they funnel information through a series of steps to a protein that's called LuxO. And it's going to run the quorum sensing show. And so all you need to know for today's talk is that at low cell density, so when the autoinducers are not there, these 3 receptors are kinases. They autophosphorylate, they send phosphate to LuxO. So in the low cell density state, LuxO is phosphorylated, it's active, and it represses quorum sensing. So in that case, the cells are dark, right? Low cell density, LuxO is repressing quorum sensing. The cells are dark, and they are not making public goods, right? They don't want to, they're alone. Right? But then what happens is as the bacteria grow and divide, when these autoinducers accumulate, they bind their receptors, right? So autoinducer 1 binds LuxN, autoinducer 2 binds LuxPQ, and that CAI1, the molecule, binds CQSS. And so when the autoinducers bind their receptors, it flips a switch. And the receptors change from being kinases to being phosphatases. So instead of pumping phosphate into the system, they pull it out. So phosphate gets drained from LuxO. When it's not phosphorylated, LuxO is dead, so it can't repress. And so what happens is quorum sensing comes on. So what that means for Vibrio harvii is that the bacteria make light, because they make luciferase, the enzyme that makes light, and they make all these public goods and release them to the environment. Mm-hmm.
Do the Luxes all act on the same side?
I don't think so.
They do.
They do.
There are many bells and whistles that are buried under this arrow about how that happens, but in the end, LuxO has one phosphorylation site, and they're all funneling information to the same protein. Uh-huh.
What's the logic of this circuitry? Is it and or?
It is an AND gate because, right, they're kinases and phosphatases. So if this molecule isn't there and this one is here, this system is pumping phosphate in and this one is pulling it out, right? And so it's a race, and they all 3 have different strengths, of each of those reactions, right? And so there's sort of why am I telling the mathematician? It's a complicated computation, right? But this signal wins, right? The me signal is the strongest. Like my vote counts more than yours. That's the way we think about it. But there's a lot of feedback loops too. Like, so for example, if they detect this autoinducer, they put more receptors on the membrane so that they pay more attention to it and they expand the dynamic range. So there's some twists in it, but in the end, you cannot turn quorum sensing all the way on or all the way off unless all three autoinducers are there or all three autoinducers are gone. Okay? Okay, but for today, for the next little while until the next question, right? What I want you to remember is that low cell density means kinase and light is off. High cell density means phosphatase and light is on, right? That's going to be the key to every experiment that I show you. Okay? All right. So now, right? So as you can see, and as you guys are asking, right? There's all these blends of molecules. These bacteria have to be able to somehow, interpret the information that's encoded in these molecules, right? And so we're really interested. I don't actually know the answer to your question, right? But so we're really interested in how this computation happens. And so what I want to talk about today exclusively is how does it happen with this molecule and this receptor. So this is the Vibrio harvii intraspecies communication molecule, and its receptor LuxN. And then the fantasy is that it'll work similarly with these others. Okay, and so here's the problem. So this is the Vibrio harvii autoinducer. I showed you that. So 4-carbon chain with a hydroxyl group on carbon 3. So the problem is that Vibrio harvii, I already told you this, it lives free living in the ocean with all these other Vibrios, right? They make mixed biofilms, they see each other, and all these other Vibrios in the ocean have their own me signal. Right? So this is Volnificus, Angularum, fischeri, right? So they're all just a little bit different in these carbon side chains. So the first question we had is, does Vibrio harvii get all mixed up, right? Does that LuxN receptor, can it tell this molecule from those, right? And if it can do that, how, right? So, or does the bacteria not really care who's out there? So we wanted to understand that question. So Xiaobo Qi, who was a graduate student that just graduated, decided to take this on as his project for his thesis. And so the first thing that he did to begin to ask how does Vibrio harvii distinguish among these very similar molecules is he built a reporter strain to detect the molecules. So what he did was he built a Vibrio harvii strain that only has the first system. So those other receptors are gone. Okay, so they're deleted. So we're not going to be complicated by those. And then he knocked out the autoinducer synthase, right? So it makes no endogenous signal, but we can make all these in a test tube. So we can add signal when we want, and the only receptor is this one. So now what you're going to remember is that a reporter strain, before we do anything, remember, because it doesn't make autoinducer, it's locked in low cell density mode, right? So that means kinase and light is off. Right? But now, of course, I just told you we can make this autoinducer. So if we make the Vibrio harvii autoinducer, remember I'm going to call this autoinducer 1, and Xiaobo just adds it with a pipette, we can trick the cells into thinking they're at high cell density. LuxN will detect the autoinducer, change into phosphatase, and the bacteria turn on light. Right? And so that's basically what happens in real life. We're just doing it synthetically. But then what we can do is ask with all these other similar autoinducers, right? We can make those as well and add them instead and ask, does LuxN get confused if you add a non-cognate autoinducer, right? So I'm going to use a color code throughout. The orange receptor likes the orange molecule. A blue molecule is a non-cognate autoinducer, right? So it's a super easy question. We just ask about light production, right? Okay. So he does that experiment. And so now what you have to do is to look a little bit more closely at this chemistry. There are 2 places that autoinducers differ. So among naturally occurring autoinducers, at the carbon 3 position, there can be a hydroxyl group. That's what Vibrio harvii has, an oxygen or a hydrogen. And then the other thing is that the chain length can go from 4 carbons. The longest one that's been shown so far is 12 carbons. Okay, so those are the only 2 features that are different on autoinducers. Okay, so we can make a whole bunch of these. Laura Miller, who was a chemist, a postdoc in the lab, she made a big collection of homoserine lactones, and now Xiaobo is going to try them with that reporter strain. So for the rest of this talk, all we're measuring is light. So this is relative light units. It's light per cell, right? And then we can add different things. So if we just add the solvent, so that's the control, right? teach the mathematicians last night what a control was. If we add, that's the negative control, right? So no light turns on. Then if we add the Vibrio Harvii autoinducer, so 3-hydroxyl-C4, that's the positive control. Here's what you can see. So see, now you're a card-carrying biologist. So anyway, yeah. So now you can see that light turns on 10,000-fold, right? That's what's fantastic about bioluminescence. Okay, so this had to work. And now what Jabot can do is add all of the molecules that Laura made and ask, does light ever turn on? And so what you can see is if we change the length of the tail, the receptor does not react. If we change what's on the carbon 3 position or the length, right, to remember you can have an oxygen or a hydrogen, what you can see is that the receptor is perfect. That's what you're supposed to see, right? Vibrio Harvii doesn't get confused because the receptor does not elicit signal transduction unless the bona fide autoinducer is provided. Okay, so that's the answer to the question about this business of these blends of molecules, is that you evolve a perfect receptor and the bacteria don't get confused. So now the question for us was, so he thought he was done, but the question was, well, how the heck's to do this magic, right? How in the world can this receptor distinguish between these molecules, right?
One of them seems, the C4HSL seems lower than your control.
Yeah, this is, well, yeah, the bar, yeah, it's yeah, I'm going to come back to that in a little while. Yeah, I think that might be right, you know, and so and this is really down in the noise for us, even though we do have error bars, you're right. You know, and so I don't know if that's true or not. I'm going to come back to something else about these in a minute. But you're right, that makes it look like it makes it more of a kinase. Right, and that might be right, and I'll show you something that's related to that, and that's supposed to be the surprise. Anyway, thanks a million. I'm on slide 5. Yeah.
How come the units for these relative units start at 10 to the 4th? Why is that?
That's the noise. So this is per some like 10 to the 8th cells. It's basically they're dark, right? It's the relative light units is per. I think it's 10 to the 8th. I can't remember how many cells, but they're dark, right? Okay. All right, so the point for now is that the receptor can absolutely distinguish between these molecules. Okay, so then the question's how, right? So the problem, right, is that we don't know very much about this receptor. So this receptor, right, it's a receptor that sits in the bacterial membrane, right, and then we can use like a threading algorithm, and it has 9 transmembrane domains. Right, and then inside the cell is this kinase and phosphatase enzymatic domains. And so the problem with receptors like LuxN, and these kinds of receptors are widespread in bacteria, right, is that we don't have crystal structures of these. They can't be purified. You can't get them in a test tube because they have this really greasy, amorphous transmembrane domain. So they have been very refractive to doing sort of traditional biochemistry. So our understanding of the receptor is this cartoon. Right, so we don't have a clue where the autoinducer binds. We don't know anything about it. You know, the guess is that it binds in the membrane domain, right? Because it's sitting out there, right? And so because we weren't going to be able to like get a crystal of the autoinducer bound to the receptor to find out how, to have a sort of atomic level look at that, Jabot wondered if he could use a genetic strategy to begin to understand how the receptor identifies the autoinducer and then distinguishes between the others. So the strategy that he decided to do was genetic, was to make a mutant library and see if he could find mutants that can't respond to the autoinducer, right? Broken ones. So here's the strategy. He cloned LuxN into a vector, right? Just the transmembrane region, so the region of DNA that encodes the 9 transmembrane domains. And then he mutagenized it. He just randomly mutagenized it. Then he put all of those together, and he made a library of mutants that have mutations somewhere in that transmembrane domain. And then he put the receptor back together, right? And then he put it into Vibrio Harvii, right? And so then the logic of it, right, is there's a mutation somewhere, right, where these X's are, in this transmembrane spanning region, right? And so then the question is, can he screen through all those mutants and look for a guy that now, if we synthetically add that autoinducer 1, it doesn't turn on light, right? So that should pinpoint mutations in the transmembrane domain that now can't respond to the autoinducer, right? And then everybody else turns on light just like normally, okay? So he did that experiment, and sure enough, he could get that kind of mutant. So now this is the one that's percent bioluminescence, right? So the wild type turns on light 100%. And what you can see is not every mutant he got was broken, but a bunch of them are dark. right? So they don't elicit signal transduction in response to the ligand, right? So now he can take those mutations and map them to that cartoon. And so what he found, which was kind of compelling, was that all of the mutations that are autoinducer blind, right, cluster to one little region, to a few transmembrane domains. And what you have to see, right, so this is the periplasm, this is the inner membrane, right, this is the periplasmic state. So they're on the outside in the periplasm, right, and so that's where the autoinducer would be. So what I think he found, right, is that this is the autoinducer binding site, that presumably somehow these couple transmembrane domains fold up, right, and the autoinducer slots in there and then changes the conformation of the protein to make it be a kinase or a phosphatase. So that was actually kind of interesting that we think we could, even without having crystal structures, we could pinpoint the autoinducer binding domain. And so that's good, except it still didn't tell us. So now that's about binding a homoserine lactone, but it still didn't tell us how does the receptor distinguish between these features, which I've shown you the receptor can do.
Know which domains are outside.
Oh, so there's program. So you can do things like you can make enzymatic fusions. We have done this to enzymes that only work in the cytoplasm and only work in the periplasm, and you can prove this topology. And then there's sort of threading domains that show outside membrane, helix, inside, right? And so we've done both of those things. So we are confident of the actual topology. We just don't have a crystal structure of it. But there's sort of standard ways to show inside, outside.
Maybe I'm asking his question again, but it sounded like you're making mutations. You're changing, I would assume, the DNA, but somehow you know that these changes correspond to the outside versus the inside. I didn't.
We do.
The mapping between the DNA.
Right,
and the
right. So you can map exactly. We know which 3 letters in the DNA encode this amino acid, this, right, yeah, right. And so this thread follows the DNA, right? We know exactly what the DNA encodes.
This picture is actually a structure in the membrane.
It is not. This is drawn by Bonnie Bassler with a pencil, right? And so
It's a physical structure.
It does. It represents a physical structure, right? So the membrane is a lipid bilayer, and this threads its way through, and this is very common for receptors. And there are ways to show, like we can make a fusion to an enzyme, like here, like this is a prediction, to an enzyme that only works in the cytoplasm. And if we make it here, the enzyme is active. If we make the fusion there, the enzyme is inactive, right? And then we have the reverse. We have enzymes that only work in the periplasmic space, and we make a fusion there, it'll be active, here it will be inactive, right? And so we have proved this structure. So there's both a predictive thing that's just computer, and then there's an enzyme thing that we did. And so this is our closest idea of what it looks like.
How many?
There's 9 transmembrane domains in this.
How many amino acids?
Oh, these are about 20 or so amino acids, which is the thickness of the membrane. There's about 20 in each of these. This whole thing is about 380 amino acids, and then there's another 400 amino acids that inside the cell that have the enzymes in them. Okay? Okay, all right, so whatever, we'll get as far as we can. Okay, so, right. Okay, so now we want to understand. So maybe we understand where the autoinducer binds, but now we want to understand how does it distinguish between the features on these autoinducers, right? So what he thought, then what Jabot thought is he could push this strategy further with those mutants. Remember, he's made these mutations all throughout, and he wondered, could he get mutants that instead of just being blind to the autoinducer, have a different specificity? Like, can you get a mutant that will actually turn on light to a non-cognate autoinducer, right? So he's going to do this experiment twice, one for the moiety on the 3rd carbon and one for length. Okay, so here's the logic. So you know in his reporter strain, there's no autoinducer there, right? We made a mutant. So this wild type Luxen is a kinase, light is shut off. I've shown you on that very first bar graph, if we add a non-cognate, right, a blue autoinducer, right, the receptor doesn't elicit signal transduction. So that I showed you. The receptor's perfect. So the question is, can he find one of those mutants that he made that now if he adds this, they turn on light, right? So they're altered specificity mutant. So if he finds such a thing, then he can go back and just ask, well, do you still respond to the cognate one or not? Right? So that's what he's going to do. And so in the experiment, right, you'll remember he's made all these mutations throughout that predicted transmembrane region. And now he's going to add blue autoinducers, so a wrong autoinducer, and ask, does anybody turn on light? So everybody gets the experiment, right, that he's going to do? OK, so twice, one for C3, what's on the C3, and one for the length. So first I'll show you this carbon-3 business. All right, so remember, the Vibrio Harvii molecule has this hydroxyl group here, and so we just Laura made a molecule that had a hydrogen, and I showed you the wild-type receptor doesn't respond to that. Okay, so he fishes through all these mutants and just looks for a guy that turns on light when you give the green autoinducer. Okay, and so just to prove to you that, again, you've seen these data in the bar graph, but here's a dose response curve. So this is the wild type receptor, that's the ligand, and you see it turns on light this is how much molecule we're adding. It turns on light in response to the cognate ligand. And it doesn't turn on light until you get to ridiculously unphysiological levels to this molecule. So that's kind of what I showed you in that first bar graph. Jabot looks for the mutants, and sure enough, he can find a mutant. So the histidine at 210, so this is your question. We know what amino acids are where. If you change that to an asparagine, now what you see is you have a receptor that actually prefers the wrong molecule to the cognate molecule, right? So indeed, you can make a one amino acid change, and you can change the specificity of the receptor. Okay, so now I showed you that molecule. This is the real molecule, and there's these three choices, so we can just test what about that new mutant, right? And so this slide is really busy, but it's actually really simple. So remember, the wild type receptor doesn't respond to any non-cognate ligand, right? I've shown you that. This mutant that I showed you liked the green ligand, it actually likes anybody. So it doesn't matter whether you have an hydroxyl, a hydrogen, or an oxygen on position 3. This mutant it doesn't discriminate what's on that carbon. And it turns out if we change that amino acid to anything else, it also doesn't discriminate. So what this result tells us is that in the wild type receptor, having a histidine at that position says you must be a hydroxyl group and nothing else is tolerated, right? Its job is to be a gatekeeper and to say, when the autoinducer slots in, to do the check, what is on that C3, and I'll let signal transduction happen or not, right? So the question then, everybody gets that? So that's the job of that. And so the question then is, well, where is that histidine 210? And of course, we can map these exactly. And so Jabot looked in the receptor for where the site is, and it's right in the middle of this ligand binding, autoinducer binding region. It had to be, or else he was fired. Right? I mean, if this is where the autoinducer binds, it better do the check for that C3 somewhere in the ligand binding domain. And so he gets to stay in the Bassler lab, lucky him. Okay, so you guys, so now we're learning something a little bit more about how these autoinducers interact with, I think, with the receptor. Okay, so here's the second time. So now he can do the same trick for the length of the tail. Right, so you'll remember that the real autoinducer has 4 carbons, so Laura made one with 8 carbons. We just doubled it. We could have made whatever length, right? So we made 8 carbons. Here's the usual control. The wild type receptor responds to its normal ligand, and I've already shown you this, right? There's no response if you change the tail length. So Jabot looks through his mutants, and sure enough, he can find one mutant, leucine at 166, that if you change it to alanine, now it actually prefers the non-cognate autoinducer over the real one. Okay? And so then we could again flesh this out a little bit. So now you have to, I want to think about how this really works, right? So leucine is a big amino acid. It likes a ligand with a tiny tail. Alanine is the smallest amino acid, and it likes a ligand with a long tail, like a puzzle, right? Like puzzle pieces fitting together. So what Jabot did was ask, is that really how this works? And so what you can see is that the wild type receptor has a leucine, a big amino acid there. And I've already shown you this. It doesn't respond to anybody longer than C4. Alanine, the smallest amino acid, will respond to C4, C6, C8, and a little bit to C10, right? So it's kind of lets any length in. If you make this is like the mama bear, the papa bear. If you make an amino acid that's a medium size, that's valine. Now what you can see is that it can respond to C4, C6, and a little bit of C8, but not the longest ones, right? So we really do think this is like a tunnel, right? And the job of this leucine is to sterically restrict anybody longer from getting in, right? So that's how we think that works, okay? So, yeah, okay? Okay, so that showed us that this leucine that's at this position, its job is to confer specificity for the tail length. Okay, so where is that? Same business, had to be, right? So it's also in this ligand binding domain. Okay, so now what we think happens, the way we really think this works, is that these orange residues fold up to make a site that accommodates a homoserine lactone. And then these two blue and green residues, when those autoinducers try to get in there, the green one says what's on C3, and the blue one says how long is the tail. And if you pass the two tests, signal transduction happens. And so then he could do a ridiculous trick, which to ask, do these guys act together or do they act independently, by making a doubly changed molecule. So what, right? So we have a molecule that's changed at C3, changed in length, and so he can make a molecule that now has the change at C3, so a hydrogen, and has a C8 tail. And this is going to be obvious how this works. If you look at the histidine mutant that is indiscriminate for C3, it can't respond to the long molecule. If you look at the mutant that likes long, any size change, it can't respond to the altered C3. If you put both mutations in the receptor together, then you now get a receptor that responds to this. So it really is fitting like locks and keys, right? And each of these amino acids does a single job, right? Which is to ask about these decorations on the tail, and they don't work together. They work independently, right? So you have to pass two tests to let signaling happen. Okay? So that's how we think it works. And so now, though, we could go back, and remember, I've shown you this slide before. We know all these other Vibrios. We know what ligand each of them uses for its intra-species communication, right? So I've been talking about this one. So these guys all have receptors, LuxN receptors, and they are in the database, the sequences. We can like actually ask, well, what do their receptors look like? If we know these rules about how it works, let's take Vibrio fischeri. So what you might notice here is Vibrio fischeri uses a C8 with a hydrogen here. That's the molecule I've been using. There's a reason we picked that molecule. It wasn't like, oh, let's just do C8 and hydrogen. We did it because we knew about this, right? So now we can actually just look in the computer at this guy's LuxN and say, what's there, right? So Jabot did that. And so what you'll see is, so this is the Vibrio harvii molecule, the Vibrio fischeri molecule. These are the real molecules they use. And so what you're going to remember from my talk is that the chain length amino acid, 166. In Vibrio harvii, there's a leucine there. That says you have to be a small autoinducer. Vibrio fischeri, that has an 8-carbon tail, has an alanine there. Remember, that's the smallest amino acid that lets big ligands in. Same thing, histidine 210. Remember, that's the amino acid that says you have to have a hydroxyl group here. In Vibrio fischeri, there's an asparagine there. And you'll remember that's the mutation that Jabot got that said you can be a hydrogen. So the truth is that Vibrio fischeri is the exact mutant that Jabot got in his screen. So you could have said, why didn't you just make that? Right? Like, why'd you go to all this trouble and why are you torturing us like this when you could have just looked in the database and seen that? But you'll remember that when we started, we didn't know even where the autoinducer binding site was, and we didn't know those gatekeeper residues. So in defense of poor Jabot, right, it only took him a year to do this. It took Vibrio fischeri 4 billion years to decide to make that mutant in order to, learn how to discriminate, right? And so we didn't have a clue. We just had that, cartoon. Right? But of course now, sorry, I'm going to get you, we can go in and look at all of these and do exactly this.
So if you compare the amino acid sequence for the two, presumably they differ in more places.
Yes, so there's many, yes, thank you very much. There's many polymorphisms throughout. Right, thank you, that's what I was supposed to say. But they made me work too hard yesterday. I had to give 2 talks and eat a lot of meals. Anyway, so they have many, many polymorphisms. So you can't just, we couldn't even line them up and actually ask, where's the homoserine lactone binding site?
I think my question, again, was similar. These are already known as separate species, so there's other distinguishing features between them, I assume, than this analysis of these receptors.
Yeah, but this
were different before.
Yeah, but this is the only thing I care about. But yes, they're very different. They have all kinds of different genes.
They're different because they switched. They got a longer tail.
There's many differences in their genomes, right? But it could be that speciation is about whether or not you can understand one another. But no, but there's
maybe this is coming up later on, but these particular molecules seem like they go in a progression, and one would imagine we go from one to the next to the next to the next evolutionarily. I don't know if that's reasonable enough.
I want to argue that in about 5 slides, right? Which is, I might add, the other surprise in this talk. Anyway, yes, I think that might be true. Like you could imagine, you start C4, you go to C6, C8, and maybe length away has to do with whether you're my first cousin, my second cousin, or
In
right.
species, do we have no sort of the age? Do we know which one?
Yeah, a little bit. We do know that. The problem is that there's many more Vibrios than people have discovered the autoinducers for. That's the problem, right? But there's actually better work in Staph aureus about for sure the autoinducer and the receptor change co-evolve. They have to, otherwise it's gibberish, right? And that with your question, I think that it is not done well, right? And especially about these weirdo glow-in-the-dark bacteria. These are not well studied bacteria that lots of people they do now work on them, but so that we know those kinds of things. But I think things like that could be done. It's not done well.
If leucine is the only responsible residue for the chain length, if the fischeri has aniline, does it respond to C10?
See, so I knew I shouldn't linger on this slide so long, right? So I'm gonna rephrase your question. That's an awesome question, right? So I'm gonna rephrase that. Remember, leucine is non-discriminant, right? It says in Vibrio harvii, it says any length can come in. So your question is Vibrio fischeri a sloppy receptor? It turns out, so Jabot asked that question, and it turns out that Vibrio fischeri, right, has a third amino acid that discriminates against short. Vibrio harvii doesn't need that, right? But Vibrio fischeri had to, and so its receptor is also perfect, but there's a third amino acid, it's in that orange spot, that Vibrio harvii does not have that, right? And it restricts against short. And so if you're going to evolve to get to like a long molecule, then you have to have a new feature to make sure you don't get confused by a short one. And that's what Vibrio fischeri did. Yeah, awesome question, right? Okay, guys get it? So they are each specific. Okay.
Aside from these amino acid sequences that you've seen to govern specificity, is there a lot of conservation in the proposed amino acid autoinducer binding site?
In the orange ones, you mean? Like in the orange? Yeah, no, they're like now when we look at them closely, there's still tons of polymorphisms, but it's sort of hydrophobic for hydrophobic, right? But you still could not have picked out. So first of all, there's only 8 or 10 luxens. It's not like you have 100 or 300 of these to do that kind of analysis, right? And there's a lot of polymorphisms, right? Like for example, we cannot simply, and it's because we cannot simply change this helix for one in Vibrio fischeri and swap specificity, right? But that's because of the question he asked, right? Yeah. So there's a lot of polymorphisms, but you can kind of now look at those luxens and you could do this basically, right? For most of the luxens, you'd be pretty close. Mm-hmm.
Can I ask a very elementary background question?
Sure.
Bacteria have ways of exchanging DNA. How specific is that? Is that within species?
So different bacteria, some bacteria do it and some bacteria don't, right? And then there's a certain bacteria that are very specific, like they actually have sex, right? And they exchange it through a sex pilus, and that tends to be with your close relatives, really close. And then other bacteria just take up DNA from the environment, and that tends to be very non-specific. And then they can make chimeric chromosomes. It is a good way to get genes right. And so some do it not at all, some do it very specifically, and some are really promiscuous.
Vibrios?
Not much exchange of DNA. Okay, so sort of 2 more parts to this, right? Okay, so now I hope everybody gets right that how this receptor is built, that this is about binding the autoinducer. But then there's something else this receptor has to do, which is once the autoinducer slots in there and it passes the tests, information has to come into the cell to change the enzyme activity, to say, let's do quorum sensing, right? So Jabot wondered, so no one understands in this class. So what I should tell you is there's only about 8 or so luxen, but there are thousands and thousands of proteins that are these sensor proteins that sense all kinds of different stimuli in the environment and that are attached to these kinase and phosphatase domains. No one knows, because everybody's in the same problem we are, how when some signal binds, how do you get the switch, right? Because they're never crystallized, they're never understood. And so Jabot wondered if he could use his strategy to figure out how, when the right autoinducer gets there, do you actually change something? This thing binds on the outside and something changes on the inside. So how does that happen? So he wondered, could he look through his mutants and just get mutants that are autoinducer independent? And so he thought, well, those would be the signaling mutants that have the switch flipped, right? So it's the same library of mutants, right? And the same logic, right? So you guys get this a million times. Light doesn't turn on. If we add the correct autoinducer, light turns on. What he wanted to do is just look through his mutants and find mutants that were bright, really bright, even when you didn't add any autoinducer. And those could be the switch mutants, right? So he looked through his mutants to find that. And again, this is just this light. And what you can see is he found lots of mutants that were bright. They're constitutively bright. There's no autoinducer there. Okay, so he maps them to his cartoon. And again, this worked really well. All of those switch mutants, those are the silver ones, are on the inside half of the receptor, and they're right under the ligand binding domain. So the idea then is the autoinducer slots in, the test happens, the receptor says, yes, this is the cognate autoinducer, and then it communicates by some conformational change, that's ambiguous, to these residues that then are on the inside half of the membrane, change the enzyme of the receptor, right? So we actually think we've identified the switch region, at least in this receptor. Okay, so that's how we think this works. So now we can make a model for what we think is going on. So forget about mutants. This is about how we think it works. So at low cell density, if there's hardly any autoinducer around, you want to be carrying out individual behaviors. You don't want to be spilling public goods out all over. So the receptor is a kinase. That means no quorum sensing, no public goods. As you and your kin grow, if a lot of the right autoinducer are there, it slots into that receptor, these tests get passed, and then the receptor switches into a kinase using those silver residues, and quorum sensing happens, and everybody makes these public goods, and the whole collective profits. However, if the wrong autoinducer is there, what you've seen from Jabot's work is that the receptor does not elicit signal transduction. It stays as a kinase, and so then the bacteria don't accidentally make public goods if they're not around their kin. Okay, so that's obvious, but the surprise, now I'm going to go back to the two questions that got asked me. Not eliciting signal transduction is not the same as ignoring these non-cognate autoinducers. So now I want to prove to you that this receptor absolutely pays attention to whether those blue molecules are there or not. Okay, so here's one more. This is a twist on the experiment that we've been doing. All of those blue molecules are inhibitors. They're going to be antagonists, right? They're homoserine lactones, right? They look like the right molecule. So here, I'll explain the experiment. So you've seen these two before. At low cell density, in the reporter strain, if we just add solvent, the reporter strain stays dark. If we add the cognate autoinducer 1, and now we're only adding it at the 50% response curve, so light turns on, right? If we keep that autoinducer there at that level, and now we add a non-cognate autoinducer, what happens? So if we add C6, what you can see is light turns off a little bit. So that inhibits the response to the real autoinducer. C8 a little more, C10 a little more, C12, they're dark. That's your question, right? So the further away, the longer it is, the more potent an inhibitor this molecule is. So the receptor is not impervious to these other molecules. It actually binds them, it responds to them, but they are inhibitors. And so what we think that means is that Vibrio harvii is tuned in to the other out there, right? And it does not elicit signal transduction if there's a competitor around. So the way we really think this works is low cell density, obviously you don't want to make public goods. If you're at high cell density and it's all your clones, sure, let's turn on all these public goods, let's be givers, right, and do quorum sensing. But even if you're at high cell density, if there's plenty of your kin around, if you are in fierce competition out there, and there's non-kin, even if you have the right amount of the cognate autoinducer that you should turn on quorum sensing, if there's somebody else around, that inhibits the ability of the bacterium to turn on quorum sensing. So in fact, it's sort of like cutting off your nose to spite your face. Even if you have plenty of kin around, you don't want to get cheated on. And so in fact, the bacteria do not make public goods at the exact cell density as over here if there are other autoinducers provided by other bacteria in the mix. And so we think this is a really cool way that the bacteria avoid being exploited when they're in a mixed species environment, is to allow the other autoinducers not to elicit signal transduction, but to inhibit. Okay, so that's how we think it works. And then, everything I've been showing you is with these mutants, and synthetic molecules. And so the question, and this goes back to the logic of the circuit, right, is can this really work, in a real bacterium that has the full complement of the quorum sensing circuit? So what Jabot did now is to change back to wild type Vibrio harvii. So I have only one more experiment to show you, right? So with wild type Vibrio harvii, where all three of these receptors are working, what happens if you do this inhibition trick with one of these non-cognate autoinducers, right? So this we're going to add with a pipette, and this now is just wild type Vibrio harvii. Okay, so here's the experiment. He takes an overnight culture, so it's very bright, it's very dense, right? So they're bright, and he dilutes it 1 to 1,000, right? So all the autoinducers get washed away. The bacteria go to low cell density. They're going to be dark, right? And then he's going to either let them grow, and so then they're making their own autoinducers, all three of them, and they can accumulate, or he's going to let them grow. They're making their own autoinducers, and he's going to add this inhibitor, right? So this is the long inhibitor, right? The non-cognate autoinducer. He's going to ask what happens. Okay, so this is the canonical quorum sensing behavior in bacteria. This is light per cell as a function of time, which is growth. And so what he does, right, is he takes this overnight. They're very bright, right, and he dilutes it. So at the start of the experiment, light is on. He dilutes the cells 1 to 1,000. The autoinducers disappear. So over time, light turns off, right, because they're diluting out the existing luciferase, but they're growing. And so autoinducers are accumulating. And at a particular time, which correlates with a particular cell density, those autoinducers kick in and light turns on again. So this U-shaped curve is quorum sensing. Okay, so everybody understands why we get that? Okay, so that's wild type Vibrio harvii. We have done that experiment 1,000,000 times, right? So what happens, so that's this one. What happens if that inhibitor is there in the context of all three of the circuits? So that's the red line. And so you're supposed to notice two things. So the first thing is that light goes off further, but then it does come on. So what this shows you is that even when all three systems are there, if you simply inhibit one of them, Luxen, what you can see is that the bacteria perceive that, and so they are inhibited, right? So light keeps turning off even more. But then remember, they're growing, and all of the autoinducers are accumulating. And in this case, we're just adding a single level of the inhibitor. And so what happens out here is the bacteria, I think, perceive, oh, there's some amount of competitor, so we're going to keep light off, but it's not increasing, and we're still increasing. And so then later, the endogenous autoinducers overpower it, and quorum sensing kicks in. And so presumably they have detected that they're inhibited, but if they keep growing, they're like, oh, we won. And even though there's some competitor here, now it's okay because there's so many more of us to turn on our quorum sensing. And so then they start making public goods when they keep growing. I mean, that's our interpretation of this result. And so,
any idea what the relative concentrations are?
right, so we do know how much of this, we actually know how much of every autoinducer is there, right? And we know how much of this, how many bacteria that would be, right? And so, it would be like that, I can't remember it exactly, but it would certainly be like that there are many fewer of these once you start getting out here. Like, so we added this at like 100 nanomolar, and out here, Vibrio harvii's autoinducer is probably at 1 micromole, is probably 100 times more. Yeah, it's about 100, I think, I'm not remembering that right, but I think by here, Vibrio harvii would probably have about 100 times more autoinducer 1 than what we added. But your question, John, we actually just want to do it, right? So this is the end, you guys, so I swear. So still, you're right, we just added a certain amount, right? But we can just do it by making a mix. So remember, I've told you about this Vibrio fischeri bacteria. This is an inhibitor, right? So we can just put the two strains together and let them duke it out, right? And we can measure all of those autoinducers at times. So this experiment is not done. This is the one we're working on right now. So we're just going to put bacteria that make their own autoinducers, so both autoinducers are increasing. And of course, we can add any proportion of the different cells, right? And so this is the experiment we'd love to do, right? So then you could imagine, so Vibrio harvii is supposed to be the orange, right? If there were lots of Vibrio fischeri around, right, making the inhibitor, Vibrio harvii shouldn't be bright. It should be dark. That we can measure. And then we can also ask, do you have a problem living, right? Like if you don't, who survives in these competitions if you can't do these public good production, right? And then remember, we have this Vibrio harvii mutant that thinks it's the Vibrio fischeri receptor, right? So we can put that in there and say, well, what happens to you? Sure, it'll turn on light because now it detects both autoinducers, right? But is it bad if you have a sloppy receptor and you start making all these public goods? Does Vibrio harvii get outcompeted? And so I don't know. So that's how we're going to try to actually ask the question in something that approximates a real biological situation, right? So we can both measure light in each strain, and we can measure the number of each cell that's there and ask, are you doing quorum sensing, and do you get a growth advantage or disadvantage? Mm-hmm.
Extreme, but these things can move by themselves.
Yeah.
Is there any possibility, let's say you had a concentration one in this area, they were sort of mixed, but there is some variation, there's more concentration in one area, will they be able to speak out the concentration?
Yeah, so these are all in well mixed, and everybody we're doing that deliberately because it's a much simpler experiment. They're well mixed. Everybody perceives exactly the identical environment. We do a lot of things where we have them in biofilms and in topology where there are gradients, where there's flow, and where there's, and that is a very complicated, right, the way and that we're trying to do with my friend Howard Stone, who's a fluid dynamics physicist. So we've started on that. This experiment will not be done that way because we couldn't interpret the result.
The mutants, are they still the ones which don't have the other?
No, no, so we can do it either way. So we might do it where they each only have autoinducer 1, to start, but then we can put that mutation we can just replace lux n with that one. But we might start with just only autoinducer 1, to try to make a more simple experiment first, right? So there's only orange and purple triangles, right? Yeah.
Quick follow-up on your comment about flow. Because I was thinking in the beginning, these bacteria, they rely on diffusion, but there's flow everywhere in the ocean, in the rivers, and everywhere.
Yeah, it's the ocean.
I've seen how it's done biofilms.
Yeah, I'm on those papers, FYI. right, yeah, right.
But how do they overcome this? I mean,
so
it's the molecules that they put out, it will be washed away.
Absolutely, absolutely. Okay, so the answer is going to be I don't know, but absolutely. So this well-mixed that's in Princeton, New Jersey, in an incubator, right? And so now the big question in my field is when you put time and space, in there, like how can quorum, because there's flow everywhere, it's ubiquitous. And so I think the way it really, really works is they're in biofilms, right? So they cover themselves with a scoop, they're adhered to surfaces, and there is restricted flow and restricted diffusion inside a biofilm. But remember, if you're just swimming around in the ocean, and the autoinducer is gone, you are by yourself. You should be in low cell density mode, right? And so that is exactly what we're trying to do with Howard, is to put him in contexts that more mimic the physical environment of a cell and ask what happens with flow. And we've done some things, like if we put them in a long pipe, right, the guy down here and there's flow, these guys have no autoinducers and those guys have a ton of autoinducers because they got theirs and everybody else's, right? And then they're screaming on in quorum sensing and these guys are not. And so then they have different behaviors. And so those are the kinds of things we're trying to do with Howard now, but it's important to have the molecules and have all these mutants so we can test these hypotheses. Yeah.
That the bacteria in the gut are carrying out a core function rather than having a core constant population. So my question is, do you have a conceptual framework to explain the way that bacteria might be doing, signaling each other but in the opposite way than this, in a cooperative way, which would explain why, as the population changes, it still carries out this core function, and when we remove one strain, it doesn't seem to carry out the same function as when it's in an interdependent situation with other species?
Yeah, I think that's a really complicated question, right? Because we don't know very much about quorum sensing in the gut, but I think that's a fantastic idea. So, right, they're all one-offs, right? These guys are competitors, right? But you can imagine these consortia, like your teeth, it's known too, that they, I don't want this to be altruism, but they need each other, right? Like on your teeth, the first 3 guys have the adhesin, but they don't have the enzyme to cut up all the goodies, so they need guy 4, 5, and 6. And so when you get those kinds of consortia, where, they would kill each other if they could, where they actually depend on each other, because they're like specialists. I'm the carpenter, you're the car mechanic, right? Then I think you can absolutely think about these kinds of rules, but you would have autoinducers that do flip, that aren't inhibitors, but that actually are agonists. You could imagine that, right? And so I think they're all one-offs, right? And you have to study a few of these model systems, right? But I think you just make sloppy receptors, right? Or you communicate with that universal molecule, and then, you know, if you see a lot of that universal molecule, it means somebody else is there because you don't make enough of it yourself, right? So I think there's lots of ways to begin to get at that. Yeah.
This proposed experiment, is it a time course experiment, or is it just how quick?
No, we're going to do a time course experiment. We're going to count species, and we're going to measure quorum sensing outputs.
Generation time, do you get facultative cheaters?
We will. So we have to watch for that, right? But I think that happens over a long time, right? You know, like to get cheaters that would take over the population, right? You would need some number of days, right? But we can sequence the luxens out of everybody that's in the experiment as we go, right? But you're right. We have to watch out, right, that they're not just mutants. Yeah.
Yeah. So you described two species that emit autoinducers, but you could imagine cheating by just Harvey, which just don't turn on. They just use the public goods, but don't produce them.
Are there such species that exploit other species?
Absolutely. No. So another way to think about this, right, is to say Vibrio fischeri is tricking Vibrio harveyi, right, to miscount, right?
Or Vibrio fischeri just not turn.
Exactly, right, exactly. So Vibrio fischeri also, yes, exactly. Vibrio fischeri also has quorum sensing, right? And if Vibrio harveyi isn't, and so its autoinducer, this is your question, the C4 is not an inhibitor of the receptor. It doesn't signal, but it's not an inhibitor of the receptor that uses the long tail. And so it could be that Vibrio harveyi is tricking Vibrio fischeri into making all kinds of public goods, right? And it's exploiting it. Yeah, I think these are all, you know, that it might, I don't know which of those is, but that I think maybe we'll get that. And so then you can imagine if Vibrio fischeri dies in this context, right? It's because Vibrio harveyi is cheating on it by making, like maybe you get something by making a short signal that this guy really is blind to, right?
Well, this is a very similar question about whenever there's a public good, there's an incentive to cheat and
absolutely
someone else. But I didn't know if that might also be a clue to the evolutionary history that you want to imagine a new species emerge is more likely to exploit the earlier species than the other way around.
Can be, right, yeah, you can, yeah, and so of course I don't know the answer to those questions, but the fantasy is, what? Yeah, I need a job, and yeah, and plus I got to do that flow stuff, and plus I'm trying to make a medicine, yeah, and plus I got to eat a lot of good meals and, you know, flit around, and you know, and so yeah, and so you can, I mean, the fantasy is that by doing these, and actually not just going out in nature, but doing these with these very specific mutations, and we can add and take away, you could actually begin to try to understand the order of events and what it takes. You know, that's a fantasy.
So, most of these bacteria live in this environment. And some of the autoinducers have a longer and longer carbon change, which makes them more and more hydrophobic.
Right.
So, is there a correlation between lifestyle and the length of those that live in the gut or light organ? Would not need a long chain.
Right. And so again, the ecology and the life history of these kinds of bacteria, not done. Right, but absolutely, and I want to go back. So right, so you guys get that. This is super hydrophobic. It probably doesn't. This guy with that is actually a little bit hydrophilic, and so you can imagine that they have very different diffusion properties, especially when you put them in biofilms. And then remember, there's 3 autoinducers, right? And if you actually look at the 3, this is medium in Vibrio harveyi. CAI1 is really hydrophobic, and AI2 is really polar. And so maybe, beyond number and who it is, those autoinducers say something about my chemical environment. You know, like if it can get away, like there's short range and long range signals. You know, like we don't know how to do the experiment to ask, are they extracting that kind of information? Right, like this guy only lives in a biofilm, and so it uses this long, you know, one, you know, and so, but even within Vibrio harveyi, it's got 3 extremely different autoinducers, and I wonder about that, and I don't know what to do. And nobody's done the natural history of these, like to be able to make a sensible, like reasonable answer to that question. Yeah.
I have another very elementary background question.
Okay, you can ask me that, but you guys actually do have to see the heroes, okay? And then you're not allowed to leave, okay? Because I didn't do anything. So this is, okay, and then I'll answer. This is Xiaobo, He's now a postdoc at Harvard working on the microbiome, right? He's awesome chemical biologist. And this is Laura Miller. She's a professor at in San Diego. She was the chemist. Okay, and now you have to actually see the people. Okay, ready.
So the question is, the membrane, where is that? Is that just under the cell wall, or how does it all work with the cell wall?
So maybe I should have a chalk. Maybe I'll be a mathematician. So these kinds of bacteria have two membranes. They have a membrane that's around the cytoplasm that has the DNA. Then they have a little periplasmic space, and then they have an outer membrane, right? And right, so they have compartments like.
They don't have a cellulose cell wall.
Cellulose?
Or the hard cell wall.
They do. So they have two. They have a double membrane. They have an inner cell membrane, and then they have this periplasm, and then they have an outer cell membrane, right? And so we call that the cell envelope. It's not exactly a cell wall. And so this receptor is in the inner cell membrane. And so things can diffuse into the periplasm, right, to touch it, and then the fanny of the receptor is in the cytoplasm where the DNA is.
In our cells, we have mitochondria, which
were bacteria.
Some resemblance to bacteria.
They were bacteria, yeah.
They have the same kind of membrane?
They have membranes that look more like, it's not the double cell wall, but the actual components, the phospholipids that are look more like these guys than like ours. I think that's true, yeah.
Since we got started on membranes, do you think that these different species of Vibrios, do you think that they have different pore sizes on their outer membrane so that they could exclude?
Oh, that's a fantastic idea. The autoinducers don't have to go through the porins. They actually freely diffuse across the outer membrane.
Oh,
so,
okay.
Yeah. So they're not the porin. So it's passive diffusion gets them out, and then back in to the periplasm. So the concentration outside is the same as the concentration inside. Okay. Yeah. So I don't know the answer to your question, right? About whether or not they can take up different nutrients, but we know the porins aren't required to detect the molecules.
How do you protect against intra-species cheaters? So it's cheaters that spontaneously arise through mutation that no longer respond to quorum.
Absolutely. So that right. So that
the commons.
Exactly. So so this was with Howard. We did a little experiment. Right. So you're going to get when you get tragedy of the commons, everybody loses. Right. So that's one way to avoid it. Right. And so we did this is that so that has to happen. Right, where so you get what he's asking. You just get by every time they replicate, you make a mutation. If somebody doesn't like it would be awesome. Like if I were in charge of this, I would make the autoinducer and I would never make the public goods. Right. So how do you avoid that? Right. So somehow cheating is controlled, or else quorum sensing wouldn't be here, right? And so I think part of controlling cheating is having these very specific receptors, right? And in this inhibition. But you're right, like how do you get rid of guys that got a mutation and now I'm a freeloader? And we don't know the answer to that, right? And I presume, presumably if you get to a high enough level, you can't grow on these substrates, and so that it's controlled in sort of the similar way that ecologists think about it, is that you do get tragedy of the commons, right? And then it collapses, right? But it hasn't been proven.
Or they could be the ones that generate the new autoinducer.
Exactly, they could be on their way to being a new species, right? Too, right? And like if absolutely. I mean, I presume that is what happened originally, is that this guy was that guy, and then yeah. And you can imagine that the way the speciation part, which is different than cheating, is the way you think about it is you had a specific molecule and a specific receptor, and then something changes, and you have to have a sloppy receptor in between, right? Which is like the one Jabot made. He made some sort of sloppy receptors, and then you become specific in the second step to a new molecule, right? So that, right? And like the way that Jabot did that, and then Vibrio fischeri gets a way to discriminate, right? And so I think maybe, yeah.
Well, one issue that might be related to that, so these bacteria are producing different categories of chemicals, the species-specific and the genera-specific and so forth. Are those just being output at a constant rate, or do those change based on what the environment seems to feedback?
So, right.
It's like in more stealth mode when they're a minority or something.
So, yeah, they do that. And so remember, so there are about 25 or 30 other components under these arrows, and there's 9 feedback loops so far that we've discovered in this. And so, for example, so the reason this is called autoinduction for this guy, the the me molecule, what happens is when I flip my bit, like I decide I'm going to go into quorum sensing mode, there's a feedback loop that one of the first genes that turns on is the gene that makes the enzyme that makes that molecule. So I flood the system with molecules. So what we've shown that does is that imposes synchrony. So like, all these bacteria are perceiving, these molecules as bumping around. Somebody thinks, oh, it's time, right? And so if I make 50 times more autoinducer, all of you come with me. And so we've shown if you knock out that loop, they're really noisy. If you keep that loop, they're like soldiers, right? And so there's there's other ones like where there's feedback. This is what I told you to the membrane. So if I start perceiving this molecule, the bacteria put more of this receptor on the membrane, and that expands the dynamic range that they can perceive, and it helps them ignore those two, right? And so it's like, oh, I'm really going to pay attention to this one. Right? And so they have all of these tricks, and we don't really know what all of those things do, but we've found 9 feedback loops that make it faster, slower, noisier, and so presumably all those things are somehow playing out into this robust signaling. And this is just this one little story in that. Yeah.
So I'm wondering about the underlying physical mechanism of these specificities. So you show this biological experiments showing that. But I'm wondering, is this mostly because of the thermodynamic affinity difference between the autoinducers of the receptors, or the actual difference is small, but during the cascade process, the energy comes in, some kind of energetic process comes in and amplify this difference?
Right. So we need crystal structures. You don't have to understand the actual KDs, like the affinity of each of those autoinducers for the receptor. We don't have that, right? We have a perceived KD, which is about bioluminescence. But what we do know, right, so it's a kinase or a phosphatase. You're either turning one on or the other. The second the phosphate hits here, which is, milliseconds later, it's over, right? Once it's in, your anti is in, and this is phosphor, not phospholuxose. So all the action is there. Right, we do know that, right, from mutants, right? And so it is all about whether the affinity of the receptor for the ligand, right, and how strongly you can either turn on or off the kinase, right? That we don't understand, but once that reaction has happened, it's over, right? Yeah.
Your intrageneral molecule is an amine.
Yeah, it's really weird chemistry.
So one wouldn't expect it to be intimately stable, especially
it isn't.
In water.
That's another thing, right? So that one has a very short half-life. So this is back to your question. Besides hydrophobic, hydrophilic, right? They all have. That one is very prone to changes. It is the least stable of all of them. You're right, especially right. We found that out when we tried to purify it, right? Anyway, yeah, and so it has the shortest half-life, right? So I think that is built in. You have molecules that stay around a long time. You have molecules that go away. This one, the 5-carbon one, undergoes all kinds of cyclic reactions, and only certain moieties are bound by the receptor. And so I think you're right that all of those features are somehow giving the bacteria information, right, about time and length scales. Yeah. So that one is like, yeah. So we keep it protected, right, when we make it.
And the corresponding diketone is
not active, none, right? The diketone is a intermediate on the way to making it, and it has 0 activity. We make that one. We did, yeah.
You showed the 9 membrane crossing things. Is that 9 membrane crossing a common structure?
So they usually they can have 2. They can have 12, right? So so 9 is not uncommon or common. They usually at least have 2, right? You have to come out, put your nose out there, and come back in.
Okay. The one thing I was interested in, there's a very famous class of receptors, and also have 7.
Yes.
Have you found 7?
There are 7, but they are not. So people do not. So these are how bacteria see the world. Yeah. This kind of receptors, the way eukaryotes see the world, are with receptors that look a lot like that have 7 transmembrane domains. People do not believe that 7 transmembrane domain proteins evolved from these, which are called 2-component proteins, right? So this is the bacterial solution. Pardon?
It's basically coincidence.
It's coincidence, except that you have to solve a problem, which is to see a stimulus on the outside and transfer that information to the inside. So, but this kind of protein is how bacteria respond to their world, and 7 transmembrane proteins is the way our cells respond to the world, and they apparently didn't come one from the other.
I was just wondering, this is sort of going out on a limb. You mentioned a switch between the kinase and the phosphatase. Okay, I think kinases play a major role in cancers.
Definitely, because those 7 transmembrane proteins are,
Okay, so what I was wondering, I realize this is extreme. One of the things you want to understand in cancer is what switches a cancer from being relatively benign to going to metastasis. And intuitively, you might suspect that the logic of it is similar to the logic of a shift from the kinase to phosphatase, and something like that might happen in cancer, shifting from benign to metastatic.
So eukaryotic kinases, right? They don't have the phosphatase, so that's a very bacterial thing. So with eukaryotic receptors, it's this one-way street. You're either off or you're a kinase. And you're right, you get mutations in those and you get cancer, or it can be metastatic. But this thing of being to go both ways, kinase and phosphatase, that doesn't exist, as far as I understand it, in eukaryotic receptors. Have I exhausted you? I think so. Okay, I have to still do one more time, right? I have this fantastic lab, and these 2 people did all of the work that you said, and I just want to thank you for these very generous couple of days and letting me get to talk to everybody in Stony Brook. So thanks, you guys.